Welcome to ASCP Exam Mastery. In this video, we are going to see medical microbiology, especially the gram-positive Cocci. Gram-positive Cocci is divided into two categories based upon the catalyst. The catalyst test will be positive for Staph aureus and it will be negative for Streptococcus. And further, we can see the Coaglis test that will differentiate the Staph aureus from Micrococcus and Coaglis negative Staph. So the Coaglis positive is Staph aureus and Coaglis negative is Micrococcus and Coaglis negative Staph, and the oxidase positive is Micrococcus and negative is Coaglis negative Staph. Based upon the bacitration, the Micrococcus is susceptible to bacitration and Coaglis negative Staph is resistant to it. And novobiocin, it further divides the Staph epidermis from Saprophyticus. So Saprophyticus is resistant to novobiocin and epidermitis is susceptible to novobiocin. As we are seeing only the gram-positive Cocci, this is a small chart that only differentiates the Micrococcaceae family, but we will be seeing Staph and Streptococcus in future videos. Let us see the family Micrococcaceae and their general characteristics. So those are generally the gram-positive Cocci, aerobic or facultative anaerobes, catalase positive except Stomatococcus, and it's a part of the normal flora, occasionally pathogenic. So what is gram-positive Cocci? We have seen in the previous videos, but still it's just an intro. It appears like a small round cells in clusters or tetrads, and they retain crystal violet stain. When stained, it is like the purple color, and it is differentiated from Streptococcaceae by catalase test. What is the clinical significance of the gram-positive Cocci? Commonly, it's a part of an indigenous flora. It can be isolated from a variety of diseases. It is opportunistic infections. It will be caused in the immunocompromised host. Let us see what is a catalase test. So, catalase test is based upon the enzyme catalase. So, the Micrococcaceae family usually have catalase enzyme, and it breaks down hydrogen peroxide to water and oxygen. And the positive test shows like vigorous bubbling, and the negative test shows nothing. It's just due to the presence of oxygen. So the Micrococcaceae are catalase positive and Streptococcaceae is catalase negative. This is a key test to differentiate from the Streptococcus from Staphylococcus or the Micrococcaceae family. So as you know, the Staphylococcus is always catalase positive and the Streptococcus is always catalase negative. It's because Streptococcus or Micrococcaceae, they have the catalase enzyme present in their isolate, but Streptococcus do not have the catalase enzyme. This test used like 3% hydrogen peroxide, and a loop of the microorganism is like mixed in the hydrogen peroxide, and it will give a vigorous bubbling. You can see the bubbles in the picture here. So that shows the presence of the catalase enzyme in that species. It can be done in like a slide method or in the tube method as shown in this picture. Another important test is the Coaglis test. Coaglis test is also based upon the enzyme Coaglis, and it used to differentiate species of Staphylococcus, especially the Staphylococcus aureus and the Coaglis negative Staphylococcus. So the most prominent Coaglis negative species that is seen in the humans are Staphylococcus epidermidis and Staphylococcus saprophyticus. And Staphylococcus aureus, that is Coaglis positive, is pathogenic. Most of the Coaglis positive species are rare in humans. So what is Coaglis test and what is Coaglis? Coaglis is the enzyme that converts fibrinogen into fibrin. So in this test you may see like it's positive, the tube shows you like a clot formation, and the negative is clear. Let me explain the Coaglis test. There are two type of coagulase, that is, one is free and another one is bound coagulase. And coagulase is a clumping factor. There are two kinds of method as shown here, the tube method, and there is also a slide method. So for the tube method, it takes at least 4 hours to see the results. So you have to add like 0.1 ml of the culture to 0.5 ml of a plasma. It may be a human plasma or a rabbit plasma, and you have to incubate for 4 hours at 37 degree, and you can see the results if it is like if there is any clot formation or clumping. So any degree of clotting or clumping is positive in this test, because this is a more accurate test, and but it is a time-consuming test. And the tube method is used for the free coagulase enzyme, and for the bound coagulase enzyme is the slide method. That is a very rapid test. It is like the results can be in 10 seconds. You have to add 1 drop of the human plasma or the rabbit plasma, and then add 1 drop of the heavy suspension of the bacterial organism, and then you have to mix well, and within 10 seconds you can see the clotting or the clumping in the slides, and that is a positive test. If there is no clumping, it's a negative test. This test may be a rapid test for the bound coagulase, but it's not accurate. Let us see the genus set up same like parasiticides, but this has the hunter program and Micrococcus is resistant for this test. Okay, there is another genus known as Staphylococcus, and it is a part of the normal flora. It's rarely isolated from infections, so it's not of clinical significance. But the colonies, you can see they are like an exception in Micrococcaceae family. They have like catalyst negative, and they are their strong agar surface. So that's it for this video. For the video, I will be doing the Staphylococcus and then separate one for Streptococcus. So this is the Micrococcaceae family. So just recall once again. If you have any questions, leave me some comments. And thanks for watching. And you can subscribe for more videos. I will be doing one by one hereafter.